Hi, good evening. We will wait for maybe 5 more minutes and then I will start today's session. Okay. Good evening, everyone. Thank you for joining today in between festive modes. So today we will be summarizing week 4 lectures and look at few assignment questions. Okay. So I will start. In case someone else is joining, just unmute yourselves and let me know, because once I start presenting, I won't be able to see. Okay. My screen is visible to everyone. All right. Hopefully it's visible. I will start. So today again we will continue with uh, cloning part. Okay, basics of cloning. In the previous session we looked at the basics of cloning and what can be the different genetically modified organisms, which can be bacteria, plants, animals, or viruses. All right, so an example of the whole workflow of how to prepare a genetically modified organism, we looked at it using an example of a plant, which encodes for an insecticide, which or insecticide which kills the pests feeding on these plants. The plants uh, inherently doesn't these plants inherently don't have that particular insecticide, or don't have the ability to produce that particular insecticide which can harm these pests. However, we can actually uh, clone, or put a particular gene encoding for that particular insecticide inside the plant cells via cloning, so that it can actually, these plants can protect, these genetically modified plants can now protect themselves from pests. Okay, so now to this lecture, in this lecture videos we will be looking at the different enzymes which are used in molecular cloning. So these are called restriction enzymes or restriction endonucleases, which cut DNA sequence at specific sites. Okay. Then there's polymerase. We looked at PCR in the previous tutorial video, which is basically to amplify the number of copies of our recombinant DNA molecule. Then there's alkaline phosphatase enzyme, which removes the terminal phosphate groups or PO4 3 minus groups from a nuclear DNA strand. Then there's DNA ligase, which is responsible for joining two DNA strands. Okay, so we will look at these, especially the restriction endonucleases. Um, in general, exonucleases or endonucleases, restriction enzymes in general, and phosphatase and DNA ligase. How do these enzymes function and what are the different types? How are these named, etc. So, in general, so bacterial cells actually have these restriction enzymes, uh, were actually discovered from the natural metabolic system that is present in bacterial cells, where they actually defend themselves from viral attacks or any other pathogens. What actually happens is, there are, suppose this is an example of one restriction enzyme, which is EcoR1, which has a specific site where it can act. Okay, if the DNA strand has this particular sequence GAATTC, then it will recognize this strand and then cleave in between these two nucleotides G and A. All right. Now what happens is, if any kind of foreign DNA is entering, pathogenic DNA is entering the bacterial cells, what the system does is, um, it actually methylates its own, uh, like methylates the host DNA All right, the bacterial cells own DNA is methylated. Either it can be methylated on both the strands or hemimethylated. Okay, so what happens is the methylated ones, the methylated due to the methylation of nucleotides at specific nucleotides, what happens is these enzymes recognize specific sequences. So due to the methyl groups present at certain nucleotides, these restriction enzymes aren't able to really recognize the specific site of action or site of cleavage. So the bacteria's own, like bacterial cells own DNA is protected. However, the unmethylated DNA, which is actually from the pathogenic source, that gets cleaved or degraded. All right. This is the basic mechanism of how a restriction methylase system works. Okay. Um, So, few things, few terminologies to remember. Uh, this is a term called palindrome, where which means that it reads the sequence. These sequences, the nucleotide sequences, actually reads the same from 5 prime to 3 prime direction on complementary strands. This is suppose 5 prime to 3 prime. This is 5 prime to 3 prime direction, right? So this is reading as TTA GCAC. The same one from 5 prime end on the complementary strand also reads exactly the same. Okay, so this is called a palindromic sequences or a palindrome. And another is mirror repeats. What happens here is it is on the same strand, but it looks as if you place a mirror over here, it will look as if it's exactly like a mirror image of this particular sequence, right? If a mirror is kept here, it will read the same as CAC GATT CAC GATT. Okay, these are like completely two mirror images of each other. Okay, these two sequences. So these are called mirror repeats. Now, whenever any restriction enzyme acts at its specific cleavage site, what happens is it can generate two types, broadly two types of ends. One is sticky ends, one is blunt end. Okay, so in sticky ends also there can be different types, where sticky ends basically is a double stranded DNA which is um, cleaved at specific sites, having few nucleotides protruding. What happens here is the lengths of the two strands, the two complementary strands that have been cleaved, can be the same or need not be the same. All right. So what can happen is, suppose this is the parent strand or the original DNA strand we are talking about. Now it has been cleaved by a certain restriction enzyme. So it can either produce a 3' cohesive ended DNA, or the 3' cohesive ended means 3' cohesive ended means here or here, um, like some more nucleotides can add over here, right? Also or it can also be called as 3' protruding ended, because these 3' ends are actually protruding out, okay, out of the um, double stranded DNA fragment. Or it can also be called as 5' recessed ended. That means there is uh, mainly the 5' end is shorter comparatively. All right. If we look at, like, this is a very generic way of saying. However, if we look at the nicked double stranded DNA, what happens is 5' end, 3' end is longer, 5' end is shorter. So it can also be called as 5' recessed end or a 3' protruding end. Okay. Or it can be the other way around, where the 5' end is actually protruding. It can be called 5' protruding ended double stranded DNA strand, or 3' recessed ended. Okay. So these are basically sticky ends, where few nucleotides are actually matching. Few nucleotides are matching and few are missing. Okay. However, blunt-ended DNA strands, this is the original DNA strand, double-stranded DNA strand. Now the length of even after the restriction enzyme cleaves this DNA strand, the length of the two strands are same and completely base paired. Here, these portions do not have any complementary base pair, right? But however, when blunt ends are produced in the double-stranded DNA, what happens is all the on both the strands, all the nucleotides are completely base paired. Okay. Now, um, there are two terms we have to be careful while using them. One is a nick, N-I-C-K, nick, or a gap. Now, when do we say that DNA is nicked? What happens here is there is absence of one phosphodiester bond in any one of the strand. Then we say that that is a nicked double-stranded linear DNA. All right, and there can be multiple nicks. That means multiple uh, places or multiple sites where one phosphodiester bond is absent in between two nucleotides. Okay, but it can be either on one strand or on the complementary strand. Anything is possible. However, it should not be on in between. It shouldn't be, like continuous or like it shouldn't be continuously absent. Then at that point of time, when more than one phosphodiester bond continuously is absent on a particular strand, then we call it in the same strand, then we call it a gap. Okay. Nick means only one phosphodiester bond is absent. Okay. However, if more than one phosphodiester bonds are actually absent continuously in the on the same strand, then we call it as a gap. Okay. Now, how exactly do these restriction modification system work? Okay. So restriction enzymes are categorized based on their structure and function. Predominantly, these are of R, S, and M types, which actually are named upon the type of function they perform. Okay, in the restriction modification system, R stands for restriction. Here, R, S, M, three components of the complete restriction modification system. R stands for restriction, where this R component is responsible for cleaving DNA at specific sites. Okay, and the S component of this restriction modification system determines the recognition sequence for the restriction enzyme, because restriction enzyme has to, recognize the specific site where it can actually sit on it and then cleave. Okay, and then there's modification system also. This component actually adds methyl groups to the host DNA, what we were talking about initially, as a like bacterial defense system. It what happens, the M component of the restriction modification system actually adds methyl groups to the host DNA to protect it from being cleaved by the restriction enzyme. Okay. Now this is the same thing that has been explained a little more in detail. What happens is restriction enzymes have specific recognition DNA sequences, like specific site where these enzymes will be acting, specific sites as in specific nucleotide sequence. Okay, and to rapidly identify the site, enzymes actually can scan that stretch of DNA via a non-specific interaction. Okay, it won't be like really tight or very specific, or so that the enzyme isn't able to dissociate at all from the site, because first it has to search for its own site of action. Okay, so it does that initially, and then the enzyme does not make interactions. So during this process, the restriction enzyme actually does not make any interaction with the bases. Instead, it makes contact with the DNA backbone, sugar phosphate backbone. Okay. And then once it recognizes the specific nucleotide sequence where it can cleave, like its cleavage site, what happens, it has to bind to that particular region. All right. Now, once the recognition site is located, enzymes make specific. Now, the interaction becomes specific, where it can interact specifically with the nucleotides that are present at that uh, cleavage site or recognition site via entry through the major groove. What are the predominant types of forces acting during that time? What kind of bonds are formed? One is hydrogen bond, and van der Waals interactions, van der Waals bonding play crucial role in this particular step where binding to the recognition site happens for the restriction enzyme. Okay. And then finally it acts on it, the closed complex that is formed between um, formed by the restriction modification system. What happens there is now it shows its cleavage activity. It actually cleaves at the specific site, resulting in fragments of double-stranded DNA. Okay. Now, this distinction we talked about, that via methylation, so as to distinguish between the self versus foreign DNA. Now, although the precise mechanism of distinction isn't known yet, but based on the literature or the studies done till now, what is suggested is that in the absence of methylation, a closed complex is formed and it allows for the proper activation of cleavage activity of the enzyme. But when a methyl group is present, either it's hemimethylated, that means only methyl groups are present on particular nucleotides on only one strand, or both the complementary sites, like nucleotides on complementary strands are methylated, it's completely methylated. During that time, the presence of these methyl groups actually hinders or affects the formation of the closed complex of restriction modification system, and it cannot really form that closed complex, and so the enzyme eventually falls from the DNA, so it cannot really act and show its cleavage activity. Okay. Now we looked at one of the names of the restriction enzymes. So how are these restriction enzymes actually named, or um, how are these given the names? So these are based on from which organisms these have been isolated, and what is the order of their identification. Okay. So if we talk about the enzyme EcoR1, so E stands for Escherichia, the genus of the species. CO comes from coli, a specific species name. Then R comes from the strain, and then 1 refers to the order of identification in the bacterium. Okay. Then there's Hind3 enzyme, HIND. Okay. Then there's BAMH1, BAMH1. Okay. So H again refers to the strain, and Hind3. Then there are many, like not one. All these enzymes are there which have been isolated from, which have been named actually from the species or organisms from where these have been isolated. Okay. Now, there can be different types of restriction enzymes, and restriction enzymes actually vary in their structure, recognition site, their cleavage site or site of action, and what is the type of cofactor that is required for their activity. So there's type 1, which actually cleaves at sites quite away from the restriction site or remote from a restriction site, and it actually requires both ATP and SAM. SAM stands for S-adenosine L-methionine. Okay, it's a multifunctional protein having both, showing both restriction digestion as well as methylase activities. So these have been identified in predominantly 2 strains of E. coli, K12 and B, and type 1 enzymes have 3 subunits, R, M, and S that we read about earlier. One R1, the R subunit shows restriction digestion activity. M subunit shows methyl transferase activity so as to protect the self DNA and differentiate it from the like self and foreign DNA. And S1 helps in DNA, like S subunit helps in DNA binding. Okay. And if we were talking about remote cleavage, so the cleavage can be between 400 to 7,000 base pairs away from the actual site of recognition. Okay. So the recognition of, then comes type 2. The recognition of type 2 enzyme is 4 to 8 nucleotide long, and it cuts the DNA within the specific site. Okay. And due to this feature, type 2 restriction enzymes have application in genetic engineering for cloning purposes, because um, the first one that we saw, the type 1, it's, suppose what we saw earlier is EcoR1, recognizes the sequence GAATTC. Okay, and it recognize and cleaves at like in between the recognition sequence only, it cleaves at in between G and A. Okay, of GAATTC. Uh, However, type 1 restriction enzymes aren't like that. Type 1 is like it will suppose it will recognize the complete sequence GAATTC or something else, suppose, but it will cleave at a position around 400 to 7,000 base pairs away. So it's variable. It's not really very specific. However, these enzymes, the type 2 restriction enzymes, are very specific regarding the cleavage site, and it is again composed of three subunits M, R, and S. And there can be different types of type 2 again based on what their function is, how many subunits these are made up of, and uh, what type of cofactor are they using, etc. Okay, so these are again various types of type 2 restriction enzymes. Now there's type 3 restriction enzymes as well. The recognition site has two separate non-palindromic sequences arranged inversely oriented, and the cleavage site or the cutting site is actually nearly only 20 to 30 base pairs away from the site of recognition. And again, type 3 enzyme is composed of two subunits, res and mod. The mod subunit is required for modification, mainly methylation of the host DNA, and res subunit cuts the unmethylated DNA. Okay. Now, when we are talking about enzymes, so in general, for cloning purposes in the laboratories, while doing your experiments, you need to add enzymes for multiple experiments at multiple steps. Okay. Now you want to, during your cloning, you want to digest a DNA. So while addition of enzymes in the reaction mixture, we have to be careful about how much enzyme are we really adding. So um, enzyme vials that come already from like company vials that come in the labs, these are like one can also purify because enzymes are basically proteins. So these enzymes can be purified as well, or they can be bought from the company as well. Okay. So barely these enzymes are used in quantities as low as zero point 0.2 microliter, zero point 0.5 microliter. Even in at such a small amount, these enzymes can actually show very robust cleavage activity or catalytic activity. Okay, so how are these enzymes actually denoted? How is the quantified? So it's called enzyme unit or U, okay, which is micromole per minute, which is with one U actually stands for the amount of enzyme that catalyzes the conversion of 1 micromole of substrate per minute. That's why this unit under the given assay conditions. Now, assay conditions is suppose assay conditions can vary depending on what is the buffer, what is the substrate, what is the buffer, pH of the buffer, how much of the salt concentration is this enzyme, like what is the optimum salt concentration for the optimum activity of this particular enzyme. All these have to be taken care of. What is the optimum temperature where this enzyme can show its maximum activity. Okay, and how much substrate should we be adding for the enzyme to actually act optimally, not not really showing proper digestion or like restriction activity, or if the substrate concentration is too much, then it can show it won't really be enough efficient in completely converting all the substrate into the desired product. Or if the substrate concentration is like suppose very low, however the enzyme has been added in the predefined unit, predefined quantity only, then it can actually over digest a particular substrate. If we are talking about suppose DNA here, double stranded DNA that we looked at in the previous slides, then if the amount of double stranded DNA added is like really less as compared to the amount of enzymes that is uh, like that is actually required according to the protocol for its optimum activity, then it can actually over digest. Where suppose we want to have, suppose this is a circular plasmid, and then this is the restriction site of one particular enzyme. Now, when it cleaves this, it will be linearized. The circular plasmid will be linearized. But if the plasmid is actually at a very low concentration, however, the enzyme unit is given at a proper prescribed concentration, what will happen is it is kind of an overdose for the reaction mixture, okay, for the substrate rather. So what will happen is it can cleave at non-specific sites also, leading to multiple fragments of the uh, double-stranded DNA. Okay. Then this is what I was talking about, the enzyme activity, which is also expressed in units per milliliter or as nanomoles per minute or per minute per milliliter. Okay, nanomoles or micromoles. And it relates actually, it relates to the concentration and varies depending on the enzyme's role in metabolic pathways, uh, DNA replication, gene expression, and many more. And there's another term that you guys might have come across earlier also, which is turnover number or kcat, which represents the maximum number of substrate molecules an enzyme's active site can convert to product per second when it is fully saturated with the substrate. Okay, so these are few terms that you might come across while reading about enzymes, enzyme activity, and all. So just keep these in mind. Okay, and your daily day-to-day purposes, even during your practical experiments and all, whenever which actually involves some kind of enzyme activity, enzyme digestion, so all these are actually prescribed, how much of enzyme units to be added in a particular reaction and all, so these have to be kept in mind. Then there's something called star activity of an enzyme. So what happens is, under non-standard reaction conditions, some restriction enzymes cleave sequences similar but not identical to their defined recognition sequence. Okay, like basically non-specific cleavage at some what I was talking about, over digestion kind of. Okay, and then factors contributing to star activity include glycerol concentration, the type of buffer, what is the salt in there, what type of pH is uh, there, how much time am I keeping the particular reaction mixture at, okay, and the presence of organic solvents. So glycerol concentration, actually what happens is, mostly all the enzymes are um, actually stored in minus 20 degrees Celsius or minus 80 degrees Celsius for day-to-day lab activities. Okay, and so these are actually kept in buffers containing high percentage of glycerol, like 50% of glycerol. What happens is then glycerol acts as a cryoprotectant. So because multiple freeze thaw cycles can be there whenever we take the thing out from take the enzymes out of minus 20 or minus 80 degrees Celsius. So this glycerol actually keeps it thawed kind of, okay, and that's how it uh, helps in maintaining the enzyme's optimum activity. Okay, so star activity of an enzyme can lead to cleavage at non-canonical recognition sites or non-specific recognition sites, or even can lead to complete loss of specificity. Okay, now enzymes, after the action of any kind of restriction enzyme, the type of how the generated ends or overhangs actually look can differ. Suppose eco R1 that we looked at in the previous examples also, it recognizes this particular recognition sequence, which is GAATTC, and cleaves at like cleaves between G and A nucleotides, okay, on both the strands. So it actually creates 5 prime overhangs, if you see here, or 3 prime recessed ends. Okay, eco R1 creates 5 prime overhangs or 5 prime protruding ends, okay, of double stranded DNA after its action. And there's another enzyme which is PST1, which generates 3 prime overhangs or 5 prime recessed ends or 3 prime protruding ends here. Okay, 3 prime OH, 5 prime phosphate, and this is the complete 3' end which is protruding, and here the nucleotides, complementary nucleotides are missing. Okay. And again, both EcoR1 and PST1 are actually generating sticky ends, not really blunt ends. Okay. So this is kind of asymmetrical, right? So both these enzymes are generating sticky ends, where EcoR1 is generating 5' overhangs and PST1 is generating 3' overhangs. Just as an example, there are many more enzymes which can come under either of these categories. Okay. So some enzymes can be blunt end generators as well, like HAE3, where it is cutting exactly between, it is recognizing GGCC sequence and cutting in between the middle G and C nucleotides. So it is generating, there are no overhanging stretch, right? What we saw earlier in the previous slide. And then there's SMA1, which recognizes triple C triple G sequence and cuts exactly at the middle, generating blunt ends. And then there's XMA1, which is actually generating overhangs, as we saw earlier. Okay. Now, restriction enzymes can have different types again based on uh, enzyme. There can be restriction enzymes that recognize the same target DNA sequence but and also cleaves it in the same way. This is called isoseiomers. Okay. Like these two enzymes, SPH1 and BBU1. Okay, it is recognizing this particular sequence and cleaving at this particular site. Then there's neoseiomers, which are enzymes that recognizes the same target sequence or nucleotide sequence. However, they actually cut at different sites. Like SMA1 actually cuts at like generating blunt ends, cuts in between C and G exactly at the middle, what we saw earlier in the previous slide. And XMA1 also is also recognizing this same sequence, triple C triple G. However, it is cutting in between the first C and C, uh, like first and second C nucleotides. Okay. Uh, Thereby, XMA1 is recognizing the same sequence, but it is generating a sticky end, whereas SMA1 is generating a blunt end. Okay. Then then there are isocodamers, which are enzymes that produce the same nucleotide extensions but have different recognition sites. Now, till now, for isoseiomers and neoseiomers, isoseiomers are cleaving the same DNA at the same DNA sequence, recognizing the same DNA sequence, and cutting it at the same site. Neoseiomers are recognizing the same DNA sequence but cutting at different points. Now, isocodamers have different recognition site altogether. However, um, the type of extension that is produced, that is same for these two enzymes. Like for example, for BAMH1, it recognizes GGA-TCC and it cleaves between these two G nucleotides and generates these types of ends, sticky ends. Whereas this SAU3AI, it also generates, it cuts at a very different sequence. However, after its activity, after its action, what happens is it generates the same type of overhang or nucleotide extension. Okay. So then these two enzymes will be called isocodamers. Okay. Next, we talked about nick in the DNA strand, where one phosphodiester bond is absent, right? So what happens is, suppose this is the DNA strand, 5' phosphate is there, and this is another, suppose half of DNA strand with 3' OH. Now, this particular DNA ligase enzyme, what happens is it uses ATP or NAD plus as a cofactor for its catalytic activity. Okay, what actually happens is in the presence of ATP, this enzyme releases inorganic phosphate PPI and then forms an adduct with AMP and AMP and then along with the DNA with 5' phosphate. So this complete complex is formed, this adduct is formed, and then it joins with the other part of the DNA fragment with 3' OH, and then this enzyme is released along with AMP, and this phosphodiester bond is formed here where these two fragments are joined. Okay, this is what is written here. DNA ligase enzyme requires ATP or NAD plus. Ligase is processing ATP to generate AMP. Right here, thereby releasing PPI, and then AMP is making an adduct with the enzyme AMP enzyme adduct to form this ligase AMP complex. This complex is binding to the 3' and 5' of DNA bearing nick and bringing them together. And after these are brought together, AMP is released and phosphodiester linkage is formed between them, okay, to seal the nick. All right. Now there's alkaline phosphatase as well, which actually cleaves the terminal phosphate groups that we looked at earlier also. All right. And then many times what happens, we need to use a linker molecule. What happens is, suppose if one wants to clone or add something to the genomic DNA, which might be like really difficult. So what happens is, an amplified foreign DNA may have restriction sites at their terminus to give cohesive end to facilitate ligation into the vector. But in scenarios when foreign DNA is a genome product, okay, and there is like very low possibility to keep restriction enzymes at the end, restriction enzyme sites rather at the ends. So what happens is, this is the DNA of our interest, and then we have to clone a particular fragment along into this DNA, suppose, or we have to clone this particular fragment onto some other vector, suppose. Now what happens here is, this is EcoR1 restriction site, we looked at it earlier, GAATTC. Okay, now this type of linker is created, and which has restriction sites over here for EcoR1 activity, and this is now actually added or ligated at the ends of this double stranded DNA here, the gray color one that is shown here. And now what happens is, in the presence of EcoR1, this actually cuts at these specific sites. Now this is actually a blunt ended DNA. Now it has proper sticky ends. Okay. So linker molecules are short double stranded DNA which are 8 to 10 nucleotide sequence long, and has restriction sites at their ends here. And 4x, a typical linker molecule with EcoR1 site, is shown in this particular figure. Linker molecule is incubated with the foreign DNA here in this example, in this picture, and ligated by the action of T4 DNA ligase to generate a chimeric DNA. Okay, and the chimeric DNA is again digested with this particular restriction enzyme having the particular recognition sequence at the ends to generate sticky ends or cohesive ends. Okay, now it is now incubated with EcoR1 digested vector in the presence of DNA ligase to get a circular clone. This one, this is the same one that has been shown here. Okay. Now, similarly, adapter molecules can be used. Sorry. Now, similarly, adapter molecules can be used, which are again 8 to 10 nucleotide long double-stranded molecule with flanking DNA sequence to provide cohesive ends. Okay. This is then adapter example of an adapter molecule with this particular sequence here, and this is the blunt-ended DNA, and this adapter molecule is added using a DNA ligase enzyme in the presence of ATP. What we saw earlier, DNA ligase acts in the presence of ATP or any other cofactor like NAD+. And then, so this here in this example, this adapter molecule actually has OH on both sides, right? So it is ligated here. Now this blunt ended DNA has phosphate, 5' phosphate, 5' phosphate on the complementary strand, and 3' OH and 3' OH here on the complementary strand. However, the adapter molecule has both OH on both the sides, but for any kind of nucleotide or strand, DNA strand, to actually ligate with this particular segment, blunt-ended double-stranded DNA molecule, it has to have a phosphate on one end so as to form a phosphodiester bond with the 3' OH group, right? So first it has to uh, ligate. So this is ligated. However, now what happens is like all the ends have OH groups. Now kinase is what we looked at in the glycolytic cycle also. Kinases are enzymes which are responsible for adding phosphate groups, right? Now some kind of kinase, which is polynucleotide kinase, in the presence of ATP is used to add phosphate groups at the end, and thereby it is creating this kind of sticky end, and the vector has already been digested. Now this particular blunt-ended double-stranded DNA molecule, with the help of an adapter molecule, um, can be ligated to this particular vector. Okay. Now we talked about different kind, like cloning, ligating the molecule of our interest or gene of our interest into a suitable vector. Now what actually makes a vector suitable? What are the parameters for what are the characteristics of a suitable vector? So it has to have a low molecular weight or size. What actually happens is, due to its lower size or small size, a small sized vector is actually robust towards shear stress, and it's easier to handle during isolation of plasmids also. If it is a bigger one, it can undergo shear stress and get fragmented upon during the isolation process, during the multiple steps that we actually do during plasmid isolation. In addition, after ligating foreign DNA into the vector, the size of the resulting recombinant DNA will be small, and it will be easier to deliver the recombinant DNA into the host cell. So small size actually helps. Next, post entry into the host should give phenotypic changes. So there should be some kind of selective marker which actually tells us that, okay, this recombinant DNA has actually um, has actually been introduced or has been taken up by the host cell. There has to be a selectable marker or selection marker or selection stress, which can help us to distinguish between the host cells that have actually taken up the recombinant DNA and the host cells which didn't take it up. Okay. Then multiple cloning site with unique restriction site. So a short stretch of DNA on the vector or carrier molecule should contain restriction sites with possible site for insertion of foreign DNA. Okay. And again, so definitely, because what we saw earlier is uh, to generate sticky ends or between like sticky ends in both the vector as well as the gene of our interest, that has to be cloned to form the complete recombinant DNA, right? Now, for generating the restriction like sticky ends, so there should be specific, we talked about the like specific recognition sequences of all the enzymes. So similarly, there should be unique restriction enzymes incorporated into the like vector. So this is called multiple cloning site in the vectors or MCS. Okay. Also, the vector should have a high copy number. Now, once it is the recombinant DNA is actually taken up by the host cell, it should be able to multiply and produce many more copies of the plasmid. Okay, because we can get a high amount of the recombinant DNA after growing the host cells under suitable conditions. Okay, or culturing the host cells under suitable conditions. Now, overexpressing a protein in bacteria which is toxic can disturb the bacterial cellular metabolism, right? Now we have prepared a recombinant DNA. The gene of our interest is actually encoding for a particular protein that we want to purify, okay? That we really want in large numbers. However, what happens is large quantity rather, not numbers. So what happens is when many in some cases overexpressing that particular protein in the host cell, in this case we are talking about a bacterial cell, can be toxic to the bacterial cell. Okay, or it can actually disturb the bacterial cellular metabolism. So in this particular scenario, a high copy number plasmid is not really desirable because it will have multiple, it will be able to produce so many copies of the recombinant DNA, and because so many copies of the recombinant DNA molecules are produced, so it will eventually encode for the uh, protein of interest, right? Because it has that particular gene of interest inserted, right? So, and then if the protein is encoded in such large quantities, it will prove to be like really toxic for the bacterial cells. Eventually, the cells will die, can die, right? So, low copy number vectors can also actually disturb the bacterial cellular metabolism, but it is slow. These changes, the disturbance are like really slow to occur, so bacterial cells can actually have enough time to get adapted to such types of changes. Okay, it can actually have some time to really adapt itself or evolve so as to evolve some kind of mechanism to counter the toxicity. Okay, and vectors that are just for cloning purposes or for carrying the foreign DNA or foreign gene of interest, then high copy number plasmids are really desirable because eventually you will get more amounts of DNA, more amounts of the recombinant DNA. Yeah, this is the same thing that we talked about earlier. Now, there are in a minimal plasmid should have some, at least a minimum number of features. We talked about few of them, the selection marker that we talked about. We talked about multiple cloning site, and then there should be an origin of replication again, like any other replicating DNA. Plasmid DNA also needs to produce its copies, right? To provide, uh, needs its own independent origin of replication to provide replication start site to make more copies. Okay, and then selection marker that we talked about in the form of either antibiotic resistance gene. Some many plasmid vectors actually harbor antibiotic resistance gene, so when these bacterial cells are plated or grown in those culture or are cultured in mediums containing certain antibiotics, then only those antibiotic I mean, only those bacterial cells which have taken up the antibiotic resistance gene, which were able to take up the antibiotic resistance gene, then those will be able to counter the effect of antibiotics and grow nicely in the culture medium having that particular antibiotic, right? Or like this is in the form of antibiotic resistance gene. There can be enzymatic genes that can give phenotypic changes or color changes upon entry of the recombinant DNA inside the host cells. Okay. Then there should be a promoter. Plasmid replication in host is performed by the host provided proteins, for example, DNA gyrase, helicase, polymerase, and DNA ligase. However, proteins are required for conferring antibiotic resistance or enzyme use for selecting transformed host cells is present on the plasmid, and a promoter adjacent to it is required to express the genes that are present on the plasmid DNA, right? So in addition, a promoter is also needed to express the genes present on foreign DNA, right? Some kind of foreign gene that has been inserted to make a recombinant DNA. So that foreign gene is usually inserted downstream of a promoter, which actually is already present in the vector, which helps in the expression of the genes present in the plasmid DNA. So our gene of interest or the foreign gene, which is the gene of our interest, is cloned into the vector having a promoter upstream to it. Okay, so that eventually it will also help in the expression of the gene of our interest that we have put it inside artificially. Okay, now bacterial plasmids can have different forms. Okay, um, it can have usually it is a double stranded circular DNA, and it can exist in three different forms. If both the strands of circular double strands are intact like this is actually called a covalently closed circle form. However, if one of the strand has a nick, we talked about nick, absence of a phosphodiester bond, then it can acquire a conformation of what is called a open circular DNA. And many times what also can happen is these covalently closed circular DNA can coil amongst itself, forming a supercoiled structure, which is called a supercoiled DNA. Okay. This is the same thing that we talked about in the previous um, slide. Covalently closed circular double-stranded DNA, nicked circular double-stranded DNA, and this is again gapped circular double-stranded DNA, which is having multiple, like where multiple phosphodiester bonds are absent on the same strand. Here the purple one in this scenario. Okay. Now this is one of the um, first plasmids to be designed, PBr322, to have a selection marker of, this is tetracycline resistant marker and ampicillin resistant marker. Okay. And this is having a restriction site over here for the enzyme EcoR1. This is having an origin of replication as well. Okay, so what we like, it has the basic characteristics, basic features that are minimal basic features that are definitely required for a plasmid. Okay. Now another example is PUC19. It has a Laxi promoter over here, and it is the initial example of bacterial plasmid having a small size and containing a multiple cloning site over here. Okay, there is this origin of replication over here, and yeah, so this is again, this also harbors. It is ampicillin resistant. It harbors an ampicillin resistant gene also. So again, it has all the basic minimal features, definite features that are definitely required for a plasmid, for a vector. Okay. Now, this is something that needs to be understood a little carefully. What happens is you have your cells that are transformed. Like, suppose these are the nutrient medium where all the cells are plated. If we talk about the bacterial cells. Now we have tried to put in recombinant DNA inside these cells. Now, suppose what we looked at in PBr322 plasmid, it has both the genes encoding for ampicillin resistance, like resistance for, like it helps the cells to, uh, you know, thrive in antibiotic containing conditions. Antibiotics are ampicillin or tetracycline. Okay. So if none of the cells are able to grow in ampicillin or tetracycline containing medium, then the cells are non-transformed. So only those cells which have been able to like really take up PBr322 plasmid, which encodes for both ampicillin resistance as well as tetracycline resistance, then or in general, suppose if the cells are able to uh, grow in ampicillin containing medium or tetracycline containing medium or both, then we can say that the cells are transformed. Now, among the transformed cells, what can also happen is, uh, suppose we have, now we don't really know if our gene of interest has been incorporated or not. If the PBr322 plasmid has only gone inside the cells, it will still help the cells to survive in either ampicillin or tetracycline containing medium. However, if we have introduced a foreign DNA or cloned a particular gene of interest in PBr322 plasmid, what people usually do is, suppose if I have inserted the gene of interest in the tetracycline resistance gene encoding region. Okay, so what will happen is, eventually, if I am putting a stretch of DNA nucleotides actually encodes for tetracycline resistance due to the presence of the plasmid PBr322. Now what is happening is, in the tetracycline resistance gene area, I am inserting a gene of a foreign DNA of interest, right? Now that will eventually like make the cells lose the capability of tetracycline resistance because we are disrupting the gene's reading frame, right? So then what happens is now uh, but the cells will are the ampicillin resistance gene is intact and the cells will grow happily on the ampicillin containing medium, right? So if the cells which have taken up the recombinant DNA are growing on ampicillin containing medium, that means good. uh, the cells have ampicillin containing medium, the host cells are growing, that means the cells have taken up the DNA, taken up the vector PBR322. But whether the vector is actually recombinant one, which actually has a disrupted tetracycline resistance, or intact tetracycline resistance. So if the cells are able to grow on the tetracycline containing medium, then those aren't the recombinant ones, recombinant PBR322 plasmids. The ones which aren't able to grow on the uh, the tetracycline containing medium are actually the transformants with the recombinant cells, right? Because in the recombinant ones, we actually disrupted tetracycline resistance, right? So when now we have transformed the cells, host cells have taken up the recombinant DNA and they have multiplied well. Now we have to like really extract the plasmids from there. So we looked at plasmid DNA extraction earlier in detail in the earlier tutorial as well. So again, I will briefly go through plasmid DNA extraction. What happens is first you have to open the cells up to take out the plasmid DNA, and here the lysis step is more um, a little gentle and little more subtle than simply digesting the cell wall with enzymes or bashing it with glass beads. So the lysis buffer predominantly contains sodium hydroxide, a base, a strong base, and detergent, SDS in this case, which is sodium dodecyl sulfate, which completely denature the plasmid as well as the genomic DNA. Denaturation means separating the DNA, double-stranded DNA, into single strands. This is actually very important to perform. The lysis step is performed quickly because excessive denaturation may result in an irreversibly denatured plasmid. So the single strands won't be able to reanneal if we do this lysis step for a longer time. Okay, so we have to be quick during this lysis step. Then comes neutralization, where potassium acetate is added so as to renature the plasmid. Now, this is a crucial step where the plasmid DNA, which is actually very small as compared to the genomic DNA or chromosomal DNA, uh, and this actually is where due to its small size, the plasmids have the advantage to reanneal quickly as compared to the genomic DNA or chromosomal DNA. So plasmids can easily reanneal, whereas which forms the double-stranded DNA, but genomic DNA being really long, it actually takes a lot of time to reanneal fully and tends to eventually tangle so that the complementary strands remain separated. So what actually happens is we again centrifuge it to separate out the plasmid DNA and the genomic DNA. So while after centrifugation, what happens is the genomic DNA, which is usually bound to proteins, form a pellet while plasmid DNA remains in the solution. Okay. And during this time, again, we have to be um, really gentle with our handling, because if we are vortexing it, if we are mixing it very vigorously, the genomic DNA can also break, right? So if the genomic DNA or the chromosomal DNA breaks, it will eventually be smaller fragments, and then it can be comparable to the size of a plasmid DNA. So it will also eventually reanneal. The genomic DNA can also reanneal, leading to genomic DNA contamination during plasmid DNA extraction. Okay. And then you eventually purify the plasmid DNA from the supernatant and precipitate it with ethanol or using phenol chloroform. We looked at phenol chloroform extraction in a lot of details in the previous class, and you can go back to the tutorial, or a spin filter. Okay. Now Usually, after plasmid isolation, we run agarose gels, which are cross-linked polymers, and it shows different kinds. We looked at the different forms of plasmid DNA. It can be nicked, it can be linear, it can be supercoiled, which travels the fastest. And supercoiled DNA is the native DNA conformation found in vivo and occurs when extra twists are introduced into the double helical strand. And just a moment. Now, in the case of DNA plasmid preps, this superhelical tension cannot be relieved because ends of the plasmid are actually joined together. It's a circular double-stranded DNA. And when we talk about the migration, the rate of migration on an agarose gel, supercoiled DNA migrates faster than predicted in an agarose gel due to its conformation. Actually, due to its supercoiled conformation, it's actually very smaller and it can easily pass through the pores of an agarose gel. Okay. And then DNA in the supercoiled form is not easily accessed by replication machinery, definitely. So during replication, there are topoisomerases which can nick one strand of the DNA helix and relax the supercoil or superhelical tension in the plasmid, circular plasmid DNA, and which actually helps the polymerases to gain access to the DNA. Now, this is linearized DNA, which occurs when this one. Nicked one is this. Linear one, supercoil travels the fastest among these three. Then the linearized DNA occurs when the DNA helix is cut in both the strands at the same time. So the linear DNA generally migrates between the nicked circle and the supercoiled forms. It may also migrate the same distance as the nicked circle. Okay. Now, if you get linear DNA when you're hoping for supercoiled, for example, after DNA plasmid preps, it might be due to nucleus contamination or harsh treatment during purification, which means eventually that your DNA strand has been fragmented badly. Okay, like nicked means only on one strand. If there's a higher amount of linear strand present, that means there might be nucleus contamination. Nucleases basically digest nucleotide sequences, right? So alkaline lysis, what we talked about in the first method of plasmid prep, where predominantly the buffer contains sodium hydroxide and detergent. So alkaline lysis, plasmid preps disrupt hydrogen bonds by the alkaline conditions, and alkaline lysis step is too harsh. In, for example, if it is incubated for a really long time, then DNA, what we talked about, that the DNA can permanently denatured, where single stranded closed circles can be formed here. Okay, that migrate ahead of all other type of plasmids on the agarose shell. Now, there's something when we talk about recombinants, transformants that we talked about in the previous slide, there is something called alpha complementation process. This is, we looked at the antibiotic selection marker, right? So there is also something called alpha complementation process, another way to see if your host cells have been able to take up the recombinant DNA or not. So what happens in alpha complementation process is, in general, we know about LAGZ gene, operon, lacoperon in bacterial cells, right? So lactose in the surrounding triggers the LAGZ operon in E. coli, which leads to the production of this particular enzyme, which is beta galactosidase, that metabolizes the lactose and exists as a homotetramer in its active state. Right? You remember, I think, ZYA genes together in a single polycystron, uh, like back in the school, high school curriculum, we read about it. So what happens is this beta galactosidase enzyme, it metabolizes the lactose, and enzyme exists as a homotetramer in its active state, like four same subunits are actually there for its functional state. Now there is a certain stretch of uh, nucleotide which is deleted from the LAGZ operon, Now, which is the delta M15 deletion mutation? What happens is this delta M15, this particular deletion results in an omega peptide with deleted N-terminal residues. Few stretches of nucleotides are removed from the laxioperon, which actually leads to removal of N-terminal, which cannot code for N-terminal residues of the complete protein. Thereby, this omega peptide, which is formed eventually, the truncated peptide is called omega peptide, which is unable to form a tetramer, and hence this omega peptide is inactive. This has deleted N-terminal residues. Now, what we talked about the homotetrameric state is the fully active state, which has a full N-terminal fragment of the protein, the alpha peptide. Okay. Now, the alpha peptide alone or the N-terminal fragment alone or the omega peptide without the N-terminal cannot perform or cannot be active by itself. These alpha peptide and omega peptides have to be together, have to be a complete homotetramer for the enzyme to be completely active. Now, plasmid DNA containing LAGZ alpha sequence, right? LAGZ alpha sequence, which codes for the alpha peptide, is transformed into LAGZ delta M15 cells, which can code for omega peptide. Now, these two, if the LAGZ alpha sequence is transformed, it will produce the alpha peptide, and these cells are already producing the omega peptide with truncated N-terminal, right? Now, when the alpha peptide, the N-terminal part, and the omega peptide with the deleted N-terminal part are expressed together, then a functional beta galactosidase is formed, and then it will eventually be able to uh, catalyze the breakdown of lactose. Okay. The rescue of function of the mutant beta galactosidase by the alpha peptide is called alpha complementation. Okay. Now, alpha complementation is again a selection marker for recombination or recombinant DNA. Multiple cloning site is present within the LAGZ sequence in the plasmid vector. Okay, what we saw in PUC 19 also. The sequence can be nicked by restriction enzymes to insert the foreign DNA. When a plasmid vector containing foreign DNA is taken up by the host E. coli, the alpha complementation does not occur, thereby a functional beta galactosidase enzyme is not produced. If the foreign DNA is not inserted or inside the vector, or if it is inserted at a location other than the multiple cloning site, then LAGZ gene in the plasmid vector complements the LAGZ deletion mutation in the host E. coli, like LAGZ alpha uh, gene in the plasmid vector, which encodes for the alpha peptide, actually complements the LAGZ deletion mutation in the host E. coli cells, which produce the omega peptide. So together alpha and omega peptides produce a functional beta galactosidase enzyme. So another thing is blue-white screening. So screening the clones with recombinant DNA where a chromogenic substrate called X-gal is added to the agar plate. Now, if beta galactosidase is produced, beta galactosidase can actually act on this chromogenic substrate, which is X-gal, which is called X-gal, to actually break this particular substrate into an insoluble blue pigment, which is a big name 5,5' dibromo 4,4' dichloro indigo, which is a blue colored substrate and insoluble. Okay, so colonies formed by non-recombinant cells appear blue in color while the recombinant ones appear white because uh, LAGZ alpha gene and LAGZ mutant host cells. Host cells are expressing omega peptide, and LAGZ alpha gene that has been cloned can express the alpha peptide, right? Now, if the particular gene of interest has been cloned in uh, like in the region encoding for a particular peptide, for the omega peptide suppose, now what happens is that won't be able to express or encode for the particular peptide. Now alpha and omega peptide won't be able to complement each other, and functional beta galactosidase won't be there. So if the recombinant DNA has actually uh, been inserted at the right place where we want it to. So the alpha complementation won't occur, functional beta galactosidase won't be produced, and it won't be able to act on this chromogenic substrate to form an insoluble blue pigment, right? However, the ones where the LAGZ alpha gene as well as the LAGZ mutant gene are able to produce their respective corresponding peptides, alpha peptide and omega peptide, then these two will eventually be able to form the beta galactosidase enzyme, which is functional, and it can act on this X-gal substrate to form the insoluble blue pigment. Okay, now on the plate, it will act as it will have, like, there are white colonies and blue colonies. So white colonies, you know that these have the recombinant cells. And then there's IPTG, which is a non-metabolizable analog of galactose. It induces the expression of LAGZ gene. IPTG is not a substrate for beta galactosidase, but it is only an inducer. So for visual screening purposes, chromogenic substrate like X-gal is required. So this is how it looks. These white colonies are the ones which have taken up the recombinant DNA, and the blue colonies are the ones which couldn't really take up the DNA, recombinant DNA, I mean. Okay. So this is what we were talking about. The competent E cells have these competent E. coli cells have this mutation delta M15, which can encode for omega peptide without the N-terminal part of beta galactosidase, and the foreign DNA actually encodes for, this is the plasmid vector, and the plasmid vector which actually encodes for this particular LAGZ alpha stretches there, which encodes for the alpha peptide or the N terminal part, right? Now if you want to, this is the plasmid vector. Now if you want to clone or insert a gene of interest, this is the foreign DNA. Now we have inserted it. So there can be three scenarios. One is this foreign DNA is inserted within the LAGZ alpha gene, which encodes for the alpha peptide. Now, if this is inserted inside the LAGZ alpha gene, then what will happen is it won't be able to encode for a functional alpha peptide, right? Or this foreign DNA can also be inserted outside the LAGZ alpha gene, right? So in this scenario, LAGZ alpha can encode for a functional alpha peptide, as in can express the fully functional alpha peptide. Another scenario can be the foreign DNA hasn't been inserted at all, right? So in the scenario where there's no insertion, or the foreign DNA is inserted outside this particular stretch of LAGZ alpha, both these scenarios will eventually encode for a functional alpha peptide. And the competent E. coli cells are already able to express omega peptide. So these will have alpha and omega peptides together will have a functional beta galactosidase enzyme that will act on the chromogenic substrate X-gal to produce blue colonies. But if the foreign DNA is inserted within this LAGZ alpha stretch, like gene sequence, then it won't be able to encode for the alpha peptide. Alpha complementation won't be there. So functional beta galactosidase uh, enzyme won't be formed, and so it won't be able to act on the X-gal chromogenic substrate. Hence, white colonies will be produced. Okay. This is the screening part, visual screening, okay, using a chromogenic substrate X-gal. So now let's move to the question answers. So the removal of the terminal phosphate groups from DNA is done by, can anyone answer? We read about it in detail extensively in the few slides previously. Uh, you have to unmute yourselves and say, because I won't be able to look at the chat. So the answer will be alkaline phosphatase, option C, okay, which removes the terminal phosphate groups. The eco-R1 methylase adds a methyl group to the dash in the eco-R1 restriction site. To which residue does the eco-R1 methylase adds a methyl group? So we looked at this particular picture earlier. So it actually adds to the A residues. Answer will be option A, adenine residues are methylated, and then it helps in the distinction between the self and foreign DNA. The cleavage site of type 1 restriction enzymes is dash base pairs away from the recognition site. Answer is option A. We looked at it like 400 to 7,000 base pairs away. So 1000 lies in that range. So option will be, correct option will be option A. Now, which of the following subunits of type 1 restriction enzymes uh, provides specificity for the recognition site? We looked at it. The option will be option C, HSDS. RSM module. S is the subunit which um, gives the specificity for recognition site. The restriction enzyme that cuts methylated DNA is, now there can be restriction enzymes which aren't able to cut the methylated DNA. However, there are restriction enzymes which are available which cuts the methylated DNA as well. Such an enzyme is this particular enzyme DPN1. Okay, this one. The BAMH1 restriction site is, we looked at isocordomers, right, which have the enzymes, the restriction enzymes, which actually have very different recognition sequences. However, the flanking nucleotide extensions that they produce are same. We looked at SAO A31 and BAMH1. So BAMH1 has the restriction site GGA-TCC, like recognition sequence GGA-TCC. The molar ratio of vector to insert for a ligation reaction is ideally. Now, we talked about the ligation of the foreign DNA as well as the vector. Now, what happens here is it can actually range from 1:3 to 1:5 to 1:7 ratio of vector is to insert. So, what happens is in cloning applications, typically 50 nanogram of vector is recommended, out of which 25 to 30 femtomoles um, is the concentration depending on what is the size of the vector. For inserts of similar size, which is comparable to the vector, usually inserts are very small as compared to the vector. Vectors are really large as compared to the insert size. Inserts can be like really maybe 80 bases long, whereas vectors are kilo bases long. Okay, but for inserts having size comparable to the vector, 1:1 molar ratio of insert to vector is recommended. If the insert is smaller than the vector, for example, 1 kb insert has to be ligated in a 3 kb vector, a higher ratio should be used where insert ratio is like really comparatively higher than the vector. Okay, and if the small inserts are really small, and even higher ratios can sometimes be used, like 1:20. Okay, 1 is the vector and 20 times higher concentration of the insert fragment. So this is something where this is NEP calculator. If you go to the site on Google, if you search it, you just give the size of your vector, etc. It will give an like the amount of insert and vector that has to be added for ligation reactions. Okay, one you can always go back and check in Google. It will eventually come. NEP calculator. Just type NEP calculator. You will have the whole calculator. You have to put in the values, put in the sizes of your insert and vector, and then different ratios for ligation will give you the amount for each amount of each amount of vector as well as the insert for ligation reaction. Now, which of the following ORI sites or origin of replication sites allows the vector to replicate in both gram-negative and gram-positive bacteria? Now, we talked about origin of replication. plasmid DNA needs its own independent origin of replication to provide replication start site to make more copies. And the plasmids containing origin of replication from col E1 can be able to grow are able to grow in limited bacterial species like E. coli, which is gram negative, whereas plasmids containing origin from RP4 or RSF 1010 like can be are able to grow in both gram positive and gram negative bacteria. So the answer is directly here. Answer is option A, RP4. This one. A covalently closed circular plasmid, when exposed to dash, can transform into an open circular form. So, a covalently closed circular plasmid, when it is exposed to which type of enzymatic activity, results in an open circular form. Okay. So, open circular form is due to a nick in the double-stranded circular DNA, right? Now, the nick ligase joins two DNA ends. Gyrase is due to used during replication. Exonuclease is a nuclease which like removes nucleotides at the ends of the DNA. But plasmid DNA is a circular double-stranded DNA. It doesn't really have any terminal points or terminal ends. So endonucleases are the one which actually cleaves nucleotides or digests DNA fragments inside within the nucleotide sequence, not at the terminal points. Okay, so the option will be B, endonuclease. The ampicillin-resistant gene of PBR322 is derived from this one. This is the same one taken from the previous slide as well as in the lecture slide it's there. So this ampicillin resistant gene has been taken from this particular RSF2124. This has been derived from there, and tetracycline resistance has been derived from this particular plasmid. Okay. So the answer will be RSF2124, option D. Blue-white screening, we looked at it in detail. This cannot be performed with bacterial colonies transformed with recombinant. Blue-white screening cannot be performed with bacterial colonies transformed with recombinant, which these are the names of the plasmids. Okay. Now, all these plasmids, for blue-white screening to occur, it should have a LAGZ gene insert inside. Okay. Now, all these plasmids, PET26B, PET28A, PUC19 have a LAGZ gene insert. Okay. However, PBR322, this is PBR322, we don't see, it doesn't have any LAGZ gene inserted here. So, it should have the particular gene which encodes for beta galactosidase, right? Like two parts of beta galactosidase. So, LAGZ alpha gene should be there in the plasmid, but PBR322 is the very basic plasmid, first vector to be formed. So, it doesn't have any LAGZ gene insert, so blue-white screening cannot be performed. So, the answer will be PBR322. Now, the correct sequence of steps during plasmid isolation by alkaline lysis is, first you have to lyse the cells, and then the plasmid DNA will renature, then everything will be centrifuged to pellet down the genomic DNA along with the proteins, and then you precipitate your plasmid DNA. Correct option will be option B. Okay. The tetracycline resistance gene produces what actually happens here. So tetracycline resistance gene is associated with TET genes, which are involved in the active efflux of the antibiotic, ribosomal protection, and enzymatic degradation of the antibiotic. Okay. So tetracycline resistance gene produces the ribosomal protection proteins out of these four options. Right. So option A. Now there can be bacteriophage lambda-based vectors also, where these bacteriophage lambda cloning vector has a middle segment, this part here, okay, which is called insertion or excision, I/E segment or I/E region, and this particular region can be actually replaced with a foreign DNA of our interest, right? Now, these cloning vectors, this I/E region can have a foreign DNA of our interest, and then what happens is, if uh, this is removed, this has a restriction site, um, and then this is I/E region is removed. Foreign DNA is also restriction digested to produce the same type of sticky ends and then ligated. Now, if this particular stretch is ligated to the same L and R regions, then there won't be any plaque formed. However, if this foreign DNA is formed, I mean ligated properly along in the LR regions, and I/E region is removed from the complete stretch of bacteriophage lambda DNA, then there will be formation of a plaque. Okay, this left arm and right arm is L and R region. Now these are two kinds of vector names, EMBL3 vector, EMBL4 vector. Okay, and this is the central stuffer region, and this has the restriction sites over here. This is the same thing that has been shown here. Okay, now cos sites are present in, if we talk about the cos sites here, cos sites are present in bacteriophage lambda based vectors, option D. So which region of the EMBL3 vector is replaced by the insert? This is the EMBL3 vector. Which region is replaced by the insert? Option will be, correct option will be option B, the central stuffer region, the yellow region that we looked at here, EMBL3 vector, which is the I/E region, okay, which is called the central stuffer region. Okay then, this is it for today's tutorial session. Thank you for joining. I will stop sharing and we'll meet again the next Saturday. Thank you so much for joining. I will end the meeting for all of you.